Hello and welcome to today's webinar. On behalf of Drug Target Review, Eurofins DiscoverX and Sanford Burnham Prebys Discovery, I'd like to thank you all for attending. I'm your moderator, Victoria Rees, Deputy Editor of Drug Target Review. Today's speakers will be Lester J. Lambert, PhD, Research Scientist in the Nicholas Cosford Laboratory at Sanford Burnham Prebys Medical Discovery Institute, and Paul Shapiro, PhD, Group Leader, Assay and Product Development in the Research and Development Department at Eurofins DiscoverX. Following their presentation, we will move on to a live question and answer session where you can pose questions to today's speakers. Please remember you can submit questions at any point during the webinar using the questions panel situated in the menu on the right. So without hesitation, may I pass over to today's keynote speakers, Lester J. Lambert and Paul Shapiro.
Good afternoon. Today we're going to split this webinar into two parts. First, I'll give a brief introduction of cellular target engagement assays and the InCELL Pulse assay platform. And then Les Lambert will give a presentation about protein tyrosine phosphatases in cancer, miniaturizing the InCELL Pulse assay, and development of oncogenic SHP2 specific inhibitors. And this will be followed by a question and answer period. So assays for compound binding and function are both important in early discovery. Biochemical and cell-based target engagement assays both provide affinity and specificity data for compound binding to specific intracellular protein targets. Cell-based target engagement assays provide additional information about cell permeability of compounds and target binding to their targets in a cellular context. Cellular target engagement assays are useful in a number of paradigms of drug discovery. For target-specific approaches, they can be useful for confirming the hits from biochemical screens in a cellular context and giving information about permeability of those compounds to the cells. And in phenotypic screening approaches, they can be useful for target deconvolution to confirm what target was giving rise to the phenotypic signal. So I wanted to go over how a cell-based target engagement assay is developed and performed. So one of the key components of a cell-based target engagement assay is to have an intracellular protein target whose biophysical properties can be monitored as they change due to compound binding. So in this case, we need a target which we can detect the changes in the biophysical properties. So, and in order to perform these assays, these cells are then grown in tissue culture. Compounds of interest are added extracellularly. They're allowed to equilibrate with the cell cultures. And if these compounds are permeable to the cell, they will equilibrate to the intracellular space and have access to the target. And then if they bind to the target, they will change the biophysical properties, which will be measured as part of the assay readout. So these assays give information both about binding of compounds to intracellular protein targets and also about cell permeability of the compounds. So now I wanted to go more specifically into how the InCELL Pulse cellular target engagement assays are constructed and performed. Over here in the upper right part of this figure, I wanted to just introduce you to DiscoverX's platform technology, which is used in these assays, and this is called enzyme fragment complementation, or EFC. And in this detection technology, beta-galactosidase is split into two inactive fragments, a larger fragment, which is called enzyme acceptor, and then a smaller fragment, which is called enzyme donor. In this case, the enzyme donor is a fusion tag to a target protein of interest. So when these two inactive portions of beta-galactosidase recombine, they reconstitute the active enzyme, which is capable of hydrolyzing a lumigenic substrate, producing light, which can then be detected in a plate reader. So now if we go over to the left, the way the InCELL Pulse assays are created is again to express a protein target of interest with a ED tag, enzyme donor tag. In this case, it's called the EPL tag. This can be expressed either transiently or stably expressed in the cells. These assay cells are then exposed to a compound added extracellularly, and the compound's allowed to equilibrate with the cell cultures. A heat denaturation step is then applied. If compound binding occurs and stabilizes the EPL tagged target protein, we then have native protein at the end of the assay, which can complement with the EA protein that's added along with cell lysis and the lumigenic substrate, and that gives us a signal in the assay. On the other hand, if compound binding does not occur and protect the target protein, which could arise either due to an inability of the compound to bind to the target, either due to a lack of specificity or affinity for the target, or a lack of cell permeability, the target protein is not protected against the heat denaturation step, and it will sort of denature and aggregate and not be able to participate in EFC, and no light will be generated in that case. So this slide shows two examples of how InCELL Pulse assays are performed. On the left, we have what we call the thermal shift assay. And here you see graphed on the y-axis, we have the output, the light output of the assay, and on the x-axis, we have the temperature increasing from left to right. And so what you can see in the black circles, in the absence of an inhibitor, we have as we increase the heat, our target protein, and this is hypothetical data, our target protein unfolds, denatures, and aggregates. And as that happens, the assay signal decreases. Now, in the red squares, we have a curve which is generated in the presence of a single concentration of a ligand or a compound binding to our target. And in this case, you can see it actually moves the denaturation curve to the right. So the target is now stabilized against the thermal denaturation. And so we can talk the shift in the horizontal, in the temperature axis, we can call that a delta TM or a TM shift. And then the separation of those two curves in the y-axis, we can call the assay window. Now, if we can see at around 50 degrees, we do have a nice separation of those two curves. So we might pick 49 or 50 degrees to run what we call an isothermal dose response assay. So on the right-hand side, you can see we have some curves from 4 compounds. So these compounds were tested in a G9A methyltransferase InCELL Pulse cellular target engagement assay. And so this was done at a single temperature and using different concentrations of these 4 different inhibitors. And what you can see is that 3 of the compounds actually generate, protect the target protein and generate dose response curves, while the 4th compound in the green squares fails to interact with and protect the target. So here we can use this to rank order compounds and also learn about the specificity of compounds for the target. So that wraps up my introduction to the InCELL Pulse platform technology. If you want to learn more about this, please go to discoverx.com InCELL. We also have a SHP2 InCELL Pulse early access assay that you can learn more about from our CAD team. And now I'm going to hand it over to Les Lambert, who will be talking about utilizing this technology for SHP2 studies.
Thank you, Paul. I'd like to thank the organizers for inviting me today. So I'm a scientist at Sanford Burnham Prebys in La Jolla, California, and we have a program on oncogenic forms of SHP2, and we've employed the InCELL Pulse assay, and I'll tell you about that today. So we have a program on protein tyrosine phosphatases. Protein tyrosine phosphatases are critical components of intracellular signaling. They engage directly with receptor tyrosine kinases and adapter proteins for the receptor tyrosine kinases. A common misconception about tyrosine protein phosphatases is to think that they just have one function, but they actually have both positive and negative roles in regulation of cell signaling. Cell signaling stimuli, including cytokines and growth factors, are interconnected to the SHP2 protein tyrosine phosphatase regulation. So our focus is on SHP2, also known as PTPN11. SHP2 is involved in several critical pathways that are important from a medical standpoint. The RAS, RAF, ERK pathway, PI3 kinase, AKT pathway, and the JAK-STAT pathway all involve SHP2 in its regulation. The important thing to remember is that when you have dysregulation of protein tyrosine phosphatases, this is actually now recognized to be a hallmark in cancer. And so we actually have a program directed against SHP2 as it is an important contributor to cancer. So SHP2 is a interesting protein. So I'm going to tell you about its function from a functional standpoint. SHP2 is comprised of 3 domains. From the N-terminus, there are 2 tandem SH2 domains, and then there is a catalytic domain, the so-called PTP domain. Upon receiving activation signals and engaging with binding proteins, the SH2 domains actually bind to phosphotyrosine residues, and this exposes the catalytic domain to allow it to dephosphorylate substrate proteins. So in this open engaged state, this is the functional form of SHP2. In certain leukemias, you can actually have mutants that actually disrupt this closed auto-inhibited form and leave SHP2 in a constitutively active form. This the phosphatase in this form is then actually a driver mutation in some cancers. But there's another mechanism that's also important in cancer. If you have anomalous upregulation of the binding proteins, basically this will also leave SHP2 in an activated state, and this means it will be catalytically active and again help drive cancer. So this all can be understood from a structural standpoint. Also importantly, there's been a good deal of attention on the development of SHP2 inhibitors for cancer, and the most important one is currently in clinical trials from, I think, 2 companies. And so shown here are some of the details, and I'll sort of walk you through. So on the left-hand side, we have the crystal structure of SHP2 in a bound form. So I have indicated in blue and then in green the first and second SH2 domain, and then the PTP domain in orange. And the catalytic site is actually wedged between the PTP domains and the SH2, so literally blocking it. So a compound was identified by Novartis several years back, a compound which then was advanced to become a tool compound, SHP099. And the crystal structure, if you go to the next panel, you can see how it actually sits inside and in a cleft that is wedged between the SH2 domain and the PTP domain. This so-called allosteric site targeted by the SHP2 class of compounds, they refer to this actually as molecular glue. And this compound actually is then able to stabilize wild-type SHP2 in its closed auto-inhibited conformation. However, there are several mechanisms for what can happen, but basically, if this equilibrium is perturbed too much, say by the too much binding protein, this class of compounds actually can't engage with the open active form of SHP2. And so the take-home message also is that the mutants of SHP2 are resistant to the binding of this class of allosteric molecule. And there's a need, and that's what drives our program. So shown here is an example of what I'm talking about. These are actually, this is from a recent publication of acute myeloid leukemia. And basically shown here are driver mutations for AML. And you can see how SHP2 is involved. It's actually got a greater importance than just gain of function mutants for the reasons I already outlined. So people have been working on trying to develop drugs for protein phosphatases, tyrosine protein phosphatases for many years. And there are several challenges that are commonly encountered, and you have to look at the past to understand them. The first and most important challenge is selectivity. So the catalytic domains of the protein tyrosine phosphatases are actually highly homologous, especially in the catalytic site. So this means that if you develop molecules that bind to the catalytic site, you can get high potency, but you might find that they actually suffer from selectivity in being able to discriminate one phosphatase from another. The other is the bioavailability challenge. Namely, many molecules that have been advanced are phosphotyrosine mimetics. And although these can have very high potency, they suffer from the fact that they don't have drug-like properties, and so you can't advance them to become a real drug. And so these two challenges have plagued the field for a long time, and you have to recognize this when you're working on this. So here's an outline of our strategy and our program. So we're looking at trying to identify small molecule inhibitors of both the wild type and these mutant forms. So shown here is a sketch of our testing funnel. So what we'll do initially is we do a primary screen against the protein. We have used protein thermal shift and also phosphatase activity assays as part of our primary screen. Then you get hits. We then go through a process of hit confirmation and testing for selectivity against other phosphatases. You get dry powders, and then we even make analogs to try to confirm the hits that we're finding. But then we would actually then incorporate cellular target engagement in the next phase of the testing funnel, namely the InCELL Pulse technology that we're talking about today. And then actually after that, we would actually assess and gauge cellular potency, say by Western blot, looking at dephosphorylation of substrates. It's because you can be deceived about the mechanism in that later assay. So we actually would push cellular target engagement actually above it. The most important thing is that, before you task a hardworking chemist on working on a molecule, you should know that it has a proven ability to reach its target in cells. So Paul's already given sort of the outline of how this works. So I'll talk about mainly how this applies specifically to our project. Okay, so we basically will employ HEC293 T cells as the cell type. And then what we did was make constructs of full-length SHP2 tagged at the end terminus. We also would explore mutant forms of SHP2. So if you have a HEC293 T cell with stably or transiently transfected with SHP2, and then you incubate these cells in the presence of a compound, what you can then do is apply a heat pulse, either an isothermal or a temperature gradient, And as Paul outlined, if you have too hot of a temperature, the protein, of course, will aggregate inside the cell. But in the presence of a compound, this will actually stabilize the protein to some degree. And then due to the fact that it has an EPL tag, it can engage with the beta-galactosidase and do its coupled reporter activity and produce a chemiluminescent signal. So this is the specifics for our particular target. Okay, so from an instructive standpoint, we always think of it to compare it to protein thermal shift. So I'll dwell on this for a second. So in a protein thermal shift assay, you'll have the protein in vitro in a single well in the presence of a fluorescent dye. And then as you ramp the temperature, you basically will produce that one of these red curves. The inflection point then goes up, and then we would define that as the temperature of melting. In the presence of a compound, then this curve is shifted to the right, and there's a change in this temperature, which we would refer to as the protein thermal shift. Of course, again, this is in a single well. On the right, then, what we have here is data using an InCELL Pulse method using cellular target engagement. So the red curve is generated, I should point out, we would call this thermal profile. So the red curve, each one of those data points is actually, well, we'll do multiplets, and that's each one of these points is generated from one single experiment with cells in a well. So the red curve is vehicle, DMSO, with a particular protein, and then if you have a compound that binds to the protein in the cell, SHP2 in this cell, I'm sorry, not SHP2 in this case, but then you would generate this blue curve, a stabilization due to compound binding. So these thermal profiles are nice to look at, and you generate something analogous to a protein thermal shift. So shown here is our workflow. What we were able to take advantage of in my lab was a miniaturized form of the assay that was developed at the Prebys Center at Sanford Burnham Prebys Medical Discovery Institute. So they had actually miniaturized this for us. Also through the Prebys Center, we have the access to a liquid handler, so-called echo machine. So basically, we're able to put nanoliter volumes of compound onto 384 well plates. So that on the left-hand side, so the first thing we would do is, get plates with compounds or control and have those prepared. Then second step, then you have cells which are stably or transiently transfected. You apply them to the compounds in the wells, about 600, 700 cells per well, and we use either, say, from a 5 to a 13 microliter volume for this incubation. So then the cells incubate with the compound. The compound has a chance to go inside, 1 hour at 37 degrees. Then you take the plate, and we put this on an Eppendorf X50 thermal cycler, which is capable of both isothermal experiments, but also these gradient experiments that I'm going to tell you about. So you can examine the span of temperatures all at once. Then after that, you add the DiscoverX InCELL Pulse reagent mixture cocktail, and you allow this, we allow the plate to incubate in the dark for 30 minutes at room temperature. And then you can read out, we use a TCAN Spark microplate reader, you read out the chemiluminescent signal. Okay, so shown here is some data related to our program. So in this case, on the left-hand side, first thing you can see, okay, so we're looking at SHP2, the wild type protein. So the red curve is vehicle, DMSO. You can see a nice steep slope. This is indicative of a well-folded protein in the cell. You can see a nice temperature, 52-degree melt temperature there. And then in this thermal profile, the cells were incubated with SHP2, the molecule I showed you before, the so-called molecular glue compound, which stabilizes the closed auto-inhibited conformation. And that blue curve, you can see we have a nice indication of cellular target engagement for SHP2. You can see, though, here's some data using the same compound, SHP2. In this case, we have one of our oncogenic gain-of-function mutants, a mutant called E76K. you'll see this actually intrinsically destabilizes the protein to some degree. You can see the slope is not as sharp for the mutant form of the protein. And importantly, incubated with SHP2 at 10 micromolar, you can see that the curves are coincident. So this molecule is not engaging the mutant form of SHP2 mutant in the cell. So further data showing the utility of this. On the left-hand side, again, we're looking at SHP2 wild type. So in this case, we're looking at several related compounds. All of these compounds are in the same class of allosteric inhibitor. Two of the compounds I have indicated here, we now actually get a shift to the right, greater thermal stabilization. One of them, example 757, is from Novartis, and the other one, RMC 4550, is from Revolution Medicine. Both to a greater degree stabilize the SHP2 wild type protein. And then in the purple curve, you can see I have SHP836 at 50 micromolar. And you can see, I would call it a small but significant stabilization of SHP2. This was actually the original hit first identified by Novartis in their screen. So on the right, also going to confirming the mode of action of these compounds, we prepared a construct of SHP2 with just the catalytic domain and incubated with SHP2. You see no change. So again, this goes to the mode of action and using this cellular target engagement assay to confirm a mode of action for in this case, these allosteric molecules. Okay, so one of the things we actually did, Paul showed you before that you can get these very nice isothermal dose response experiments. And we actually played around a bit. So with the X50 thermocycler on the right, you can see, we can actually establish gradients in the long axis or short axis. And so we actually set it up that we would actually stamp out the ligands on going on the long axis, if you can see, and then the temperature gradient on the other. So then what we could do is actually do our optimization to find the best isothermal temperature to perform dose response experiments. So you could do it all in one sort of optimization experiment. So up in the upper left here, then you can see we actually do a dose response at multiple temperatures, and from this we obtain our optimum temperature. Then down below on the lower left, you can see a nice cellular dose response for the SHP2, again, directed against SHP2 wild type. Okay, so we have a program, again, directed against oncogenic forms of SHP2. And I'll tell you about two of the compounds that we actually discovered and how the InCELL Pulse assay has helped us analyze them and gauge their mode of action. So the compound on the left, SBI-221, shown there is actually its cellular, or excuse me, its in vitro potency in a kinetic assay direct against both the wild type and mutant form of SHP2. These are micromolar inhibitors. So underneath there, you can see SHP2 wild type, basically an InCELL Pulse experiment using SBI-221, again, engaging with its target in the cell, small but significant curve, but again, consistent with a micromolar inhibitor in vitro. To the right then, we have this compound SBI-668. I've indicated its in vitro potency. And then when we direct this compound against SHP2 E76, the oncogenic form of SHP2, we actually see that this compound is actually a destabilizer. So in DMSO in black, you can see the curve generated by E76K, and then in blue, we see the effect of the compound SBI-668. So although in general you don't really want destabilizing compounds when you perform thermal shift, I would actually ask you to think about the allosteric nature of SHP2, and so how these things might bind. And so we actually are interested in this compound, and it's even though it is a destabilizer. So the ability to do isothermal experiments provide something that you can actually employ in your medicinal chemistry efforts. So I'll tell you about what we're doing here. So basically, choosing an isothermal experiment, we actually stamped out, 384 well plates and I think quadruplicate, like 60 compounds at a time. What you could actually do is doing the isothermal experiment. We could actually, compared to control, we actually could test a large number of related compounds at once using the InCELL Pulse assay. So, these bar graphs, we have purple, that's actually wild type, and orange, which is E76K mutant. So again, the two compounds I just told you about, let's then look sort of at the details here. So this biphenyl pyrimidine scaffold compound, we tested a number of analogs, and what we saw was kind of interesting. So basically, so we had a certain series of compounds which actually had an acid at the R1 position, which you can see. And you can see in that circle to the left, you can see that these compounds have minimal ability to bind to SHP2 wild type in the cell and the mutant. In contrast, the tetrazole carboxylic acid isoster, they actually could engage with SHP2 wild type in the cell, minimal, I would say, going in interaction with the mutant form of SHP2, the E76K mutant. So again, this goes to that this compound then, or I'm sorry, using this technology, we're actually now being able to gauge cell penetrance. Basically, the carboxylic acid is less able to get in the cell than the tetrazole can. Then to the right, here's our benzothiophenone compounds. Basically, these are, like I said, destabilizers. They destabilize both the wild type and mutant form of the protein, but you can see we were actually able to compete several compounds against each other and find ones with greater ability to engage with the target in the cell. Importantly, on the far right, we can, for perspective, we actually had SHP0999 as control, and you can see it actually, just looking at the luminescence, it's much greater. It has much greater effect in this assay than our compounds, which are probably not allosteric, but it's a useful perspective. And then again, using this, you can see that the SHP0999 has a minimal ability to interact with the mutant form of the protein. So, then shown here are some using InCELL Pulse. Basically, we're able to gauge cellular potency for these compounds. So on the lower left, I have a titration dose response against SHP2 wild type. You can see then that SBI221 is able to engage with its target in the cell, and we get an increased luminescent signal. But SBI668, the one on the left, is not able to do much with the wild type protein. In contrast, on the right side, here we have SHP2 E76K. When we titrate in SBI221, it has a minimal ability to engage with the wild type protein, but then a significant ability to SBI668 can actually then bind to and destabilize SHP2 E76K. So what we're getting here, what we have with these early hits, is basically we are able to obtain even the selectivity that we're after against mutant form of SHP2. So I'll summarize what I've told you then about the InCELL Pulse assay. Namely, that you can use this to study SHP2 by looking at the target engagement of small molecules in the cell. Second thing to point out is that you can actually use it to discern the mode of action of your molecules. So we are able to actually confirm the allosteric mode of action of certain compounds against SHP2. Importantly, for our program, we can use the InCELL Pulse assay to identify small molecules that are engaging with the oncogenic forms of SHP2. This technology addresses key challenges in the drug discovery, namely that chemists are expensive, And you don't want them wasting time on things that are not really hitting their target in the cell. So this is a very useful thing to include in your testing funnel. Finally, you can actually use this to advance your compounds from a medicinal chemistry standpoint. You can test a number of compounds at once and help drive towards greater cellular potency. Finally, I'd like to thank all the people involved. Most importantly, Celeste Romero, who did a lot of this work. She's now in graduate school. I work at Sanford Burnham Prebys Medical Discovery Institute in the lab of Nick Cosford. Nick Cosford is a medicinal chemist who is doing translational research in an academic setting, and we have an incredible group with a wide array of talent. My partner on this program and the PI is actually Lutz Tautz, who's a phosphatase genius. We teamed up very closely with the Sanford Burnham Prebys Center with John Holleran and Edward Sergienko, who actually provided us the protocol for the miniaturized assay that I showed you here today. This program is funded by NCI and administered by Leidos, and I need to thank them, and also receive money from the NIH. I thank you for giving me your time, and thank you for the organizers for giving me this opportunity to talk about the things we've been doing.
Thank you, Lester and Paul, for your excellent presentations. So we now have the pleasure to introduce our questions and answer session, where today's speakers will answer all of your questions. Don't forget, you can still submit your questions using the questions panel in the menu on the right. Let's take a look at the first question. So the first question is for Lester, and it's, can you confirm the Eppendorf X50 can do 30C gradients in both directions, and that's columns and rows?
Yes, it can. Can you hear me? Yes, it can do both columns and rows. It's very useful for that. So I showed you a clever use of that, but there's other things you could try with that ability.
Thanks, Lester. So the next question now is for Paul. So Paul, how do you interpret destabilization of targets in the InCELL Pulse assay?
So I mean, I think that compound binding. I mean, I think first of all, it's indicative of compound binding, and I think, and so therefore, to first approximation, I think the information that we get is still useful for reflecting, on the specificity and the potency of of the compounds, and also in a cellular target engagement assay indicates that the compound has access to the target. As far as destabilization, I'm not a biophysicist per se, but I think it just indicates that somehow that compound is changing the conformation, opening the conformation, and therefore sort of facilitating the thermal pulse's ability to denature. Perhaps Lester, might want to sort of add to this, since he's actually, shown examples where one of their molecules did that to the SHP2.
Hi. So to answer that question, yeah, it definitely depends on the protein. What is the meaning of a destabilizer? So I'm talking about the SHP2 is a pretty intricate protein with a lot of moving parts. So if the data I showed, like I said, we're interested in destabilizers as well, but it depends on the protein. And when you get into this, one of the strengths of this is you're going to learn a lot about your protein.
Well, and I would add, you're also going to learn a lot about the ligands, the molecules that you're putting in there. So it is fascinating, the utility that that we can get from this platform.
Thanks both. So just a follow-up question, Paul. Does destabilization suggest target engagement?
To my mind, I would say it does, because I mean, obviously, the experiment, we're looking at the ability of a ligand relative to the vehicle to induce a change, in the assay, which is reflective of a change in the target protein. And so to my mind, yes, absolutely, it would reflect and indicate that the compound has engaged the target, which in turn, indicates specificity, affinity, and access to the target, or in other words, cellular permeability. Les, Les, would you, do you have anything to add to that?
Yeah, just to give something away. So compounds that destabilize their target are, in a sense, acting like PROTACs, right? So they're actually maybe causing the protein to be destabilized, and they could be potentially degraded. So that's something destabilizers might be doing, and we've done a lot of this. Certain compounds seem to actually destabilize their protein target in the cell, forgetting the the linker to cereblon or something like that. So think of it that this assay might be able to help identify things that are acting like PROTACs.
Great, thank you. So next question now. Can you explain how to identify targets for complex diseases such as Parkinson's disease? So that's for Lester, but Paul, if you also have anything to add to that?
So how to identify compounds for Parkinson's disease? Yeah, I don't really have an answer to that one. I'm not really focused on that.
Sure.
But the assay itself is versatile. So any protein of interest that's related to that disease might potentially be used in this assay.
Yeah, and I agree with what Lester said. I do have some experience with Parkinson's targets, but I think Lester was exactly right on in saying that, this platform is therapeutic indication agnostic. It is target class agnostic. It is broadly applicable to really any intracellular target of interest, and we've actually worked with many classes of proteins for many different therapeutic indications. So the versatility is very broad, and so I think there's no reason why it would not be applicable to Parkinson's disease, as it is for oncology, metabolic disease, and so on.
Great, thanks so much. So next question now for Lester. Can you make a link between the SHP2 and MAPK signaling pathway?
Can I make the link? Yes, there is a link. So there basically SHP2 is a regulator of that pathway. So this has been shown.
Lovely, thank you.
Using this assay, again, this particular assay, again, this is a chemical focused assay. So I would say if you have a potential inhibitor, you want to test, the effect of SHP2 inhibition on that pathway, and you had an unknown chemical. Now you can demonstrate that you're hitting SHP2, so you can be convinced that if you have an effect on the pathway, it was potentially through target engagement with SHP2.
Thanks. So, okay, next question now for Paul. Are there any compounds which can interfere with the InCELL Pulse assays to give a false positive result?
I mean, first of all, I would say that because this is a luminescent readout, it's quite tolerant. So in other words, we don't need to worry about autofluorescent compounds because of the luminescent readout. But I would say, so I don't think we're concerned about false positives. Of course, there would always be the issue of compound, target engagement of sort of potentially non-specific. But I think we really don't have much concern with that, with non-specific. I'm sorry, we don't have much concern with false positives based on that mechanism.
Great. So next question now for Lester, this one. Did you determine the structure of the compounds bound to SHP2 to identify the binding mode, and did you measure the kinetic values?
Work in progress. So we're looking at that.
No problem. Okay, so next question now for Paul. In your one-pot format, do you worry the hybrid lytic detergent would resolubilize aggregated proteins?
Well, just a point of clarification. So this is not, using the hybrid lytic detergent, but we're not using that platform technology. This is the InCELL Pulse, technology using the Eurofins DiscoverX EFC technology. But we do have a detergent. So after incubating with compound and performing the thermal pulse, we do lyse the cells and add the detection reagent so it has access to the intracellular targets. We're not concerned about it. I mean, these are non-ionic detergents. They're mild detergents. I don't think we're concerned about it resolubilizing the denatured protein. We've never really encountered that. So that would be what I would have to say.
Great, thanks. So back to Lester now. Do you ever see multiphasic melt curves, and how do you handle these? What do these mean?
Yes, we do see them. So I've shown you. So again, we've applied the InCELL Pulse to other targets, and so we have seen them. Interpreting them, I would say, is difficult. Is it compound induced, or is it the protein itself that's multiphasic? One of the things that you might consider with the assay is, again, you have to think about your protein. So in this case, I was able to show you that we worked with full-length SHP2, which has 3 structured domains in it. And we could have imagined that we might have seen, say, 3 melting curves, right? It's possible. But when SHP2 melts, it seems to melt as one single uniform, curve. But with other targets, multi-domain proteins, you might see like biphasic behavior. And this goes to the the construct choice of when you're trying to build this assay. Like I said, if you know about your protein, it helps drive the work. So I hope that's an answer.
Lovely, thanks, Lester. So our next question now is for Paul. Does the assay work well for both soluble and membrane-bound proteins?
So yeah, we typically think of this assay for soluble, typically cytoplasmic proteins, but having a prototypic target class would be kinases. But again, I said it's very broadly applicable. And of course, SHP2 is not a kinase, and yes, it does work with membrane-anchored proteins, and it can even work with proteins that are in organelles, such as the ER, and it even works with polytopic membrane proteins. We have examples of all of those. But having said that, most of the targets for which assays have been elaborated using this the InCELL Pulse platform are soluble cytoplasmic proteins.
Thanks, Paul. So another question for you now.
Yeah.
What is your experience of potency and efficacy readouts of your compounds in the InCELL Pulse assay in comparison to in vitro target engagement and potency in cellular activity assays? And do you use quantitatively or qualitatively?
So I'll comment to this, and then I think Lester might be able to add to it. So first of all, obviously, when we use this assay, we are getting information about the affinity for the target in a cellular context, and also the permeability. So so I would say that the potencies may be different than what sees in a biochemical assay, either due to permeability challenges of the compounds, and other other points, and this is obviously often seen in cell-based assays. It's quite common that compounds are more potent in biochemical cell-free assays than cell-based assays. And sort of to the second part of the question, we certainly so we do often see a right shift, and in some cases less potency in the InCELL Pulse assay platform. Certainly, these can be used for rank ordering compounds, so that's more of a semi-quantitative comparison. But we find that those potencies, or those rank order potencies, do hold with biochemical assays. Perhaps Lester has something to add to that based on his experience.
Sure. So like we're kind of doing that when I showed you that optimization experiment with the dose response curves, isothermal dose response curves, where we do them all at once. So when you generate a cellular dose response curves at different temperatures, remember the key thing is you're changing the temperature. Also, in terms of the let's call it the right shifting of the potency. We're measuring an isothermal dose response at say 50 degrees, but the body temperature is at 37. So that will tend to weaken, give an apparent weakening of that EC50 that you measure. So there's several things that sort of conspire to make the cellular potency weaker than it might actually be. However, we consider it useful. If you look at it, we put our focus on our ability to rank compounds, and that's the strength of the assay.
Lovely, thanks both. So question now for Paul. Can this technology be used for extracellular targets as well?
Yeah, we've considered that. I don't think so. I think the answer is no. I mean, this is and I suppose you could even extend the question. Could it be used for a plasma membrane protein where most of the protein is outside the cell? You certainly would lose a lot of the utility. You might get yes, you might get a readout. I think you probably would get a readout, but certainly you would lose one of the main benefits of performing this assay, namely, the be able to interrogate and learn about the permeability of your compounds. It might work for the extracellular domain of a protein or a protein outside the cell, but it certainly is not designed for that. And for that matter, we really don't have any information about that.
Thanks, Paul. So another question for you now. What would you say regarding a compound changing a protein's Hill slope in its melt profile, but still giving the same melting temperature value?
Yeah, that's a little tougher. So I suppose that would be reflective of target engagement. I have to be honest, we really haven't seen that. But I guess that would somehow, that would be, that's a bit confusing, but I think it would potentially indicate that you had changed sort of the cooperativity or the kinetic sort of denaturation without. Yeah, so that's a bit, that stumps me a little bit. I haven't seen that, and we probably would not develop that assay. In other words, we probably would be confused by that and would not find that to be something we could move forward with. But maybe Lester might have some other thoughts on that.
Yeah, thanks. So I had two little examples in the seminar. First off, I was trying to show you can definitely see a change in the Hill slope for our mutant form of the protein, and so that's different from the wild type. So that is probably indicative of that's because we can really understand SHP2 from the structural standpoint. what we're looking at is a melting curve that's a combination of several things. So we no longer have that higher level structure. So that leads to a destabilization of the hill slope. Then, so I showed some early data. It's actually, when you buy the kit, there's a control protein, MTH1. And so the melting curve of the unbound MTH1 is quite, quite not impressive. Definitely a weakly folded protein by itself, but then they have these inhibitors that greatly stabilize it, greatly stabilize the hill slope. I would just say to the whole thing that once you work on your protein of interest, you'll be confronted with these issues, and I don't think there's a simple answer.
But I thought the question was, but in the case of the MTH1, you certainly do see a right shift in the melt curve, in addition to a change in the hill slope. Would you agree?
Yeah.
So I think the question was, I thought the question was, in the absence of a thermal shift, but just a change in the hill slope, what would you do in that situation? And that's the situation where I, thought it probably would be a bit of a challenge to use that as an InCELL Pulse assay.
Yeah, we've seen that in the thermal profiles. I'm sorry, I did go off. We've seen that in our thermal profiles, and generally we say that's not target engagement. So just changes in the hill slope.
Thanks both. So question now for Lester. One of your shift curves was less than 1 degree, and you said it was significant. So what is the minimum shift, and would you run this several times?
Yeah, we run several replicates, and the significance. And we have a JBC paper where we analyze this in detail. So I direct you there. You actually get greater significance with these isothermal, that up and down chemiluminescence signal than you get from the actual curve fit left and right. So even I would, small thermal shifts are, par for the course with this particular target. But that's the start. But interestingly, though, you get pretty good significance when you just do the isothermal experiment going up and down. So maybe that's an answer. But yeah, I would say 1 degree is pretty much sharp cutoff.
Thanks, Lester. So one more question for Paul now. Can you use this assay for central nervous system targets, for example, neuronal cell lines, to look at targets endogenously expressed?
So the answer is, I don't see any reason why it couldn't be used in neuronal cell lines. This technology, what we described today, involves expressing heterologously expressing a tagged target protein, so overexpression, but we also have the opportunity, and we have proof of concept studies for doing genome editing, and that can allow this to be done with sort of expression at more physiological levels. But again, this assay platform technology does require tagging the protein with the EPL tag. So you cannot do this on on sort of endogenous proteins without inserting a tag.
Okay, thanks, Paul. So another question for you now. This assay works for large complexes. Does this assay work for large complexes where the ligand can bind in different chains?
So I think yes, it certainly could. In other words, in this case, you would be tagging one of the targets in a multi subunit complex. It certainly could. And we do have an example, sort of a variation on this theme, where we have a target protein stabilization, where we can actually show the effect of a target protein interaction with the target protein stabilizing the target, and that is ligand dependent. But I would say that we tend to have experience that this is more, we get better results often with globular proteins, and in some cases we actually use just the, if we know a ligand is binding to the active site, for example, in the case of kinases, we actually often use domains, you know, and so I think a multi-protein complex would certainly be a challenge, and it might attenuate the assay signal, but it's certainly not, it doesn't disqualify that target, I would say.
Great, thank you. So I now actually have a follow-up comment from the melting temperature question earlier. So if there's the same melting temperature value, why then are there different hill slopes? And comment that a potential guess would be a compound that's targeting a different domain. And that's for Paul.
So yeah, this one, I mean, this, I was challenged the first time to, you know, about the changes in hill slope without a change in, you know, in the melting curve. But yes, I think, you know, as sort of following up on Lester's explanation of, you know, the hill slope, a steep hill slope reflective of a well-folded protein melting, whereas a shallower hill slope reflective of a more open conformation. I suppose that yes, that it could, that a change in a hill slope without a change in the TM could reflect, might reflect binding to a different domain, and therefore, you know, changing the conformation slightly. But I guess, you know, one way that you could change the hill slope would be having antagonizing effects, because we can have destabilizing and stabilizing effects. And yes, so it might reflect a different specificity for different domains of the protein, but that's kind of a tough one. You know, and I think, you know, as Les was saying, having structural and all kinds of structural information about your target can help you interpret, you know, some of the data you're getting from this assay, because in some respects it's kind of somewhat of a phenotypic assay.
Lovely, thanks, Paul. So another question now for Lester: is there any other biological assay rather than thermal shift for SHP2 determination?
Rather than thermal shift for, you mean target identification? You mean like, you know, for example, you mean in our primary screen? I guess that's, I'm a little confused about the question.
I wonder if they mean function.
Yeah, I mean, so, you know, downstream, you can look at targets of SHP2 and look at their phosphorylation and dephosphorylation. So phosphospecific antibodies in the cell can be used to phosphospecific antibodies to SHP2 targets can be used to ascertain SHP2 catalytic activity in the cell. And so that's part of our platform. I call our testing funnel, excuse me. So I think hopefully that's an answer.
Lovely, thanks both. So unfortunately, that is all we have time for, but please join me in thanking Lester and Paul for their time today. So as you leave the webinar, a survey will appear on your screen asking you to rate the webinar. Please take a moment to provide your feedback. If now is not a good time, the survey will be sent to you shortly via email. If you could complete it when you can, we'd greatly appreciate it. So now, on behalf of Drug Target Review, Eurofins DiscoverX, and Sanford Burnham Prebys Discovery, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye.